Hi, this is Sir Mark, and welcome to another science video. You can remember the order of mitosis with the mnemonics PMAT. PMAT stands for prophase, metaphase, anaphase, and telophase. These mnemonics will help you remember the steps in the arrangement of mitosis. But what's most important is you understand what is happening in each stage and why it is important for the division of the chromosomes. Mitosis happens in body cells or somatic cells. It adds new cells during the development and replaces old and worn-out cells throughout your life. The goal of mitosis is to produce daughter cells that are genetically identical to their mother. Let's start by looking at the cell right before it begins mitosis. We know from the last video that cells in the interphase has already copied its DNA. So the chromosome in the nucleus consists of two connected copies called sister chromatids. In this picture, we can't see the chromosome very clearly because they are still in their long and stringy decondensed form. This animal cell has also made a copy of its centrosome, an organelle that will play a key role during mitosis. So there are two centrosomes inside the cell. But in plant cell, generally they don't have centrosomes with centrioles, but they have a different type of organelles that has also a microtubule organizing center that plays a similar role. In the early prophase, the cell starts to break down some structure and build up other. This will set the stage for division of the chromosomes. In the prophase, the chromosomes start to condense, making them easier to pull apart later on. A mitotic spindle begins to form in the centrosomes and move to opposite side. This spindle is a structure made up of microtubules. Its job is to organize the chromosome and move them around during mitosis. The spindle grows between the centrosome as they move apart. The nucleolus and the nuclear membrane also start to disappear. In the metaphase, the nuclear membrane has already disappeared and the chromosomes or the sister chromatids start to align at the middle part of the cell, or what we called as the metaphase plate. Metaphase plate is just a term used to describe the plane where the chromosomes line up. A microtubule is also extended to a chromosomes and attached to a kinetochore of a chromosomes. But before proceeding to anaphase, the cell will check to make sure that all the chromosomes are at the metaphase plate with their kinetochores correctly attached to a microtubules. This is called a spindle checkpoint. This will ensure that the sister chromatids will split evenly between the two daughter cells when they separate in the next step. If a chromosome is not properly aligned or attached, the cell will halt division until the problem is fixed. In anaphase, the sister chromatids separate from each other and pull towards the opposite end of the cell. A protein glue that holds the sister chromatids together is broken down, allowing them to separate. Each is now its own chromosomes. The chromosomes of each part are pulled towards the opposite end of the cell. All of these processes are driven by a motor proteins, or a molecular machines that can walk along microtubule track and carry a cargo. In mitosis, these motor proteins carry chromosomes or other microtubules as they walk. In telophase, the cell is nearly done dividing and it starts to reestablish its normal structure as cytokinesis takes place. A mitotic spindle fiber is broken down into its building blocks. Two new nuclei are formed, one for each set of chromosomes. Nuclear membranes and nucleoli also start to reappear. The chromosomes begin to decondense and return to their stringy form. After that, the cell is now ready for cytokinesis, or the division of cytoplasm to form two new cells. When the cytokinesis finishes, we end up with two new cells, each with a complete set of chromosomes identical to those of the mother cell. The daughter cells can now begin their own cellular lives and go to another cell cycle. That's the end of this video. If you have questions, please feel free to write it down at the comment box below. This is Sir Mark. Thank you for watching.